welcome to Machine Learning in the Brain, Vision Neuroscience and Vision Models. This is going to be the first lecture where we're going to speak about the specifics of the brain, how the brain processes information, specifically vision in this case, and then how models that are common in machine learning at the moment are linked to the inspiration between what happens in vision models, and also to set up some of the connections that we're going to see in some of the future papers about more direct mappings between how you can use vision models to explore vision in the brain, but also vice versa. So with visual illusions, one primary thing becomes apparent, and it's that we're not just actually recreating an exact manifestation of what exists in the outside world. Because if you look at this, you either might see a rabbit or you might see a duck. And what that shows us is because the actual stimulus that's hitting our eyes and being processed is actually the same, but our perceptions is being altered between whether we see a rabbit or whether we see a duck. This has this implies that there's a top level mechanism that's changing our perception, that there's a top level perceptual manipulation. There's a top-down mechanism that is actually being applied to the visual input in order to influence whether we see a rabbit or a duck. And there are various reasons why this might come about. And when this was first introduced in Switzerland, someone did a study where they asked people what they saw in this illusion, and they found that around Easter time people were more likely to see a bunny rabbit. And then around October, people were more likely to see a bird. So one hypothesis that was suggested was because there might be a higher prevalence of rabbits in the window for Easter, and because this is more prevalent in the environment, people are more tuned to seeing rabbits at that time of year, and therefore that was the primary response given when people were asked to say what they saw in this illusion. And then, perhaps by default, the illusion just looks more like a duck around October. I'm not sure. So transduction is the property whereby absorption of light in the eye, specifically in the retina, is then transformed into electrical signals that then reach the brain. So what we actually see here is we have an outside stimulus, here the letter A, that is then entering into the eye, and we have an inverted projection, which I'll explain why that is the case in a sec. And then this will travel down a visual tract and end up at the back of the brain in primary visual cortex, and then onto higher order areas here. But the idea is that we have light, and then light is being systematically converted into electrical activity, and that is the electrical activity of neurons communicating, or specifically like electrochemical activity that then results in us being able to, or humans are able to actually perceive stimuli or what's going on in the outside world. And this is very related to the camera obscura. So if you know or you speak Italian, you would recognize that the word camera is the word for room, and perhaps it's always been a mystery to you why exactly the word for room is the same as this mechanical device that we use to take pictures, but it's because the original cameras were actually rooms, specifically called the camera obscura. So what would happen is there would be a stimulus out in the real world, and the light would enter via a small pinhole, and that pinhole would then have the effect that only light at a very, very specific angle would come in, and then it would be projected down. And that is what gives rise to the inversion effect in the camera obscura. So here we have a tree, and we have the light rays, and as they enter in, the only the information, say from the very top here, as it goes down to the pinhole, it can only travel in a straight line, which then projects it down to the bottom, which results in this inverted image that we see. And so the eye is actually a camera obscura as well in terms of light, because we have a very restrictive pupil at the front of the eye, and this is why the information is mapped inverted onto the retina and then processed by the brain, and we actually see things then the other way around. And so radio waves are of many different types, and they're defined by different frequencies and amplitudes, which we'll see in a second. But what I just wanted to point out here was that as the frequencies along this band here change, that there is a band called the visible light spectrum, which is the frequencies, or the range of frequencies of radio waves that are result in the perception of light in humans. And there are also some comments or observations in other animals that snakes and bats, for example, have a wider range of visibility or are able to make use of more of the infrared spectrum in order to perceive lights. And there is also some interesting research about wolves and how they perceive heat, for example, which is something beyond what humans do, and whether that actually counts as sight into the infrared spectrum, or there's a different category of perception. There's unresolved debate there. So the properties of light, as I said, are wavelength, which also defines our perception of color, and also intensity. So the intensity of a waveform is its amplitude, and amplitude we can see highlighted here. It's just the let me just change color here. So if we have the crest and the trough here of a wavelength, then the distance between both is defined as the amplitude, and the rate over time in which we see one full cycle return through a crest and a trough, that defines one wavelength. And as you see in the visible light spectrum here at the bottom as well, the different wavelengths are defined by the different sensitivity to colors are defined by different sensitivities to wavelengths of light. And so here we see a schematic of what is going on in the eye. So we have the optic nerve here. The optic nerve here. And then we have rods and cones. So you might have heard of rods and cones before, but primarily rods are what are sensitive to the light-dark distinction, and cones are more responsive to color. So we have different cones, and each cone is tuned to a specific frequency, to a specific wavelength, and the wavelength then is defined to be sensitivity to color. And we also see that when you're actually focusing on a point, the very center of your fovea, your foveal fixation point, you can see in a lot of detail, and then what's going on around you and around the focal point is much more hazy. If you want to actually focus in on something, you need to focus your fixation point over to that thing, and then that becomes the center of your fovea. And this is where we have the highest density of cones, very high spatial acuity. Well, the purpose of the rods is to be able to detect, okay, if I'm focusing on something and I see something in my periphery, that sensitivity to change color isn't required there. It's just sensitivity, a light-dark distinction. You know that something's happening, and then you would turn your head and focus on what's actually going on there. So from the degrees away from the fovea, which we see here is 0, all of the cones are tuned to a very, very, very small range away from 0 degrees out of the foveal fixation point. But the rods occupy a much larger range, so 80 degrees one way and 80 degrees in the other way. And here we have the representations of the cones tied to different bands of wavelengths of light that are responsible for the perception of different colors. And here we have a depiction of the visual field. So similar to what I was saying before, we have the fovea and we have the blind spot, which is where the optic tract leaves the back of the eye. There are actually no perceptions there. This is something that the brain is aware of and fills in based on expectation. But actually there is a gap in our visual field that we don't actually perceive. Regarding lateralization of vision, you might be aware of the fact that we have contralateral processing in the brain of many different features. So for example, I'm controlling my left hand with my right motor cortex, and I'm controlling my right hand with my left motor cortex. As you can see, we have what we would say called the contralateral, which is like the different side of processing. And what happens in vision is interesting, but I think a few people might be aware of the fact that we do have this contralateral processing, but they might say that, okay, it's the right primary visual cortex at the back of the brain which processes all input from the left eye, and then vice versa, but that's actually not the case. So if you consider everything you can see right now, and you can divide it into two, so just like what's done here, we have this line going down the middle. Everything in this area here is called the left hemifield, and everything here is called the right hemifield. And what actually happens is the left hemifield and also the right hemifield projects to both eyes. So what you can see here, if I zoom in, you can see blue and red here. So what's going on is the right hemifield is projecting to the left part of both the left and the right eye. So you can see this whole area in the right hemifield is blue, and you can see we have blue here, we have blue here, and the opposite is true for the left hemisphere. This goes to then the right section of the left eye and the right section of the right eye, and then these travel together, and before they hit the thalamus, which we'll talk about in a few slides, they then become segregated in terms of the hemifield. And so it's not the case that the left eye input solely goes to the right hemisphere, and vice versa. So eyes come in pairs for most animals. This is a funny rhyme that was used to highlight the point of whether animals are prey or predators, and so it goes like eyes in front, it likes to hunt, or eyes on the side, it likes to hide. And so the idea here is that, you know, if you do have eyes on your side, you're more aware of your environments, because if you are a prey and you need to be aware of predators, you need to be able to have a wider access to your visual environment. And through evolutionary pressures, we see that different animals, whether they're prey or predators, have different positions of their eyes in order to allow them to, you know, better interact and survive in their environment. So humans, yeah, we have eyes in front, and therefore we sacrifice the backwards view. And then because we're getting a slightly different image from the left eye and the right eye, this allows us to then recreate a sense of 3D perception. And we do that via binocular coordination. So each point in the visual field then projects to points on the two retina, like we saw in the previous slide. And the information, so as the light hits an object that's in front of us, and it would bounce back from a convergent point. This happens for close objects, but not for objects further in the distance. So this is what is being highlighted here. If there is an object here and light is bouncing in, then from the same close target point as well, we have projections onto the retina, but then this doesn't happen for far objects. So we have a different perception, and it's this different pattern of what happens with far objects and near objects that allow us to build a perception of 3D. So we'll go into a little bit more details about it here. So if you see, there we have a tree, and then we have a policeman. The policeman is closer to the eyes, which are here, than the tree, and therefore the light, as it hits the policeman and then enters both of our eyes, like according to this principle here, that the difference in the projection of where the light is hitting both the retinas, it is more displaced for near objects. So this is where I can see these blue lines that come from the policeman, and they're actually, say, here and here. Whereas for the tree, for example, as the light is coming from for a further distance object away, then the projection to both points on the retina is actually closer together across the both eyes in the fovea, which is what we see here. And now we'll talk about the thalamus. So the thalamus is, in Latin it means the anteroom, or the hallway. And so the way I like to visualize to talk about the thalamus, it's like a sensory processing hub. As we have our sensory organs, so our eyes, our ears, etc., these take in information from the outside world and convert it into different signals that the brain needs to know about. So if you imagine there's a house, a large house that all sensory inputs need to go through. So we arrive at the main door, and if the person is conscious, this main door, front door is open, and the sensory inputs will go in, and then we could go to a different floor of the house, which could be represented by a different lobe, and then each door in each of the floors would represent a specific sub-area of the lobe of the brain. And we'll see a few different variants, like also in this lecture for what happens with vision, but then also in the language lesson we'll talk more about the perception of speech and cortical processing via the ears. As I mentioned before about this idea of the perceptual house, most senses need to pass through the thalamus, but there is one, the sense of smell, so the olfactory system does not go through the thalamus, and this is one of the reasons why we, if someone is unconscious, for example, when people are unconscious, it's like the doors to the sensory house in order to get to the thalamus are closed. You can't access it. If someone's unconscious, the processing pathways are blocked into the thalamus as well. So by providing a strong olfactory input via the nose, and which then doesn't need to go via the thalamus to the cortex, it can then jumpstart the brain and promote emergence out of an unconscious state, which is why these smelling salts work. But back to the idea of the thalamus. Okay, so what happens with vision? We see that light hits the back of the retina, and information is then sent via the optic tract down to the primary visual cortex at the back of the brain. But as I said before, we first have to go through the thalamus. So the region of the thalamus that processes visual input is the lateral geniculate nucleus. We have one of them in each hemisphere. And here is a fMRI demonstration of how we can detect this lateralization of inputs. So here we're actually looking down on the brain in this plot. So if you can imagine that we would have the eyes here, and this is the back of the brain in primary visual cortex. And what happens here is we present a checkerboard to either the left hemifield or the right hemifield, and we also have at the bottom here right and left, and these associated colours to show exactly how far in the visual hemifield, whether it's left or right, the exact angle away from the fovea that is present in the checkerboard stimuli. So as you can see, if what we see for left, which is these bright colours, because remember we're looking down at the brain, so this is actually the right hemisphere. So the left input from the left hemifield is actually processed here in the right lateral geniculate nucleus, and the converse is true with this blue colours here in the left hemisphere for information from the right hemifield. And back at the brain here we have primary visual cortex, which again is segregated, as we can see. So we do know from experiments like these that even though the left hemifield and the right hemifield are projected to both eyes, the segregation into specific components happens before the information is passed from the optic or along the optic nerve and before it hits the thalamus. And this is the kind of result that we can see why that is the case. On the right we see a anatomical scan of where someone's highlighted the left and right lateral geniculate nuclei, and then also the back of the brain here we have primary visual cortex. And so the tract that takes visual information from the eyes to the lateral geniculate nucleus in the thalamus is called the optic tract, and then the name of this tract that's highlighted here is called the optic radiation pathway, which takes visual information from the thalamus, specifically in the lateral geniculate nuclei, down to primary visual cortex, or V1, as we'll talk about. And here is the anatomy of a real brain showing the optic radiation pathway, and the blue arrows are also pointing out the exact location of the right geniculate nuclei. So for receptive fields, if you imagine this purple square is a TV screen that you're looking at, for example, and we're fixating on the black dot in the middle. This is our fixation point, and we're recording from one neuron in the visual system, and this neuron has a receptive field, and which means that it is sensitive to light stimulus whenever it is present in its preferred location. So if you look at this white circle in all of these, in the right plot here, what we have are recordings of action potentials over time, and often we see background firing. So it's not the case that if you're going to record that you're only going to detect action potentials when there's a stimulus in the receptive field. Background firing is a commonality, across all neurons in the brain, and it's deviation from background firing that actually carries the information that we're interested in, and it's actually the difference from baseline firing that actually carries information. So what we see when the light stimulus is in the bottom left, we have background firing here, and then when the light stimulus is moved to the closer to the receptive field, we still see about the same background firing. As the light stimulus starts to approach the receptive field, we start to see a little uptick in the rate of action potentials that we're able to record as well. So it's saying that something has been detected. And then when the light stimulus is actually directly in the receptive field, we see a very, very high rate of action potentials and like maximal firing rates in order to. It's sort of like the neuron is ringing a little bell to say, like, I am tuned to detect light at this very specific spot, and that is actually what's happening now. And it's the connection of these small receptive fields together that then lead us to be able to perceive slightly more complex objects, and this then slowly increases while we build up the complexity of the visual stimulus in order to detect the things that we can detect in the real world. But first we'll talk about primary visual cortex for a little bit. So as I mentioned before, primary visual cortex is at the back of the brain here, and you can see it's inside the calcarine fissure. So there are a few different names that people use to talk about the primary visual cortex. We can call it primary visual cortex or V1, as well as the calcarine sulcus. Typically, if we're speaking cognitive neuroscience, we would either say primary visual cortex or V1. More in an anatomical sense, where you're not specifically focused on vision, you might call it by a various Brodmann area or the calcarine sulcus. And here we see this on a actual human brain. But in the bottom image, if here we look at the fovea, and we're actually taking part of I believe it's a monkey brain, but I'm not 100% sure. But what you can see here, these numbers represent the sensitivity to angles away from the fovea. So we have all of this area here that is specifically to detect, to correspond to information that is perceived within 10 degrees left or right of the foveal fixation point, where we have the highest spatial acuity. And then a little bit later, or a little bit further along this pathway, we have about the same patch, which is to detect the other 30%. So as you can see, that there's a different amount of cortical infrastructure devoted to where we need high spatial acuity, and then where we can actually sacrifice a little bit and be less precise in terms of the sensitivity to what's going on in the environment. And that is also something we see in this idea of the sensory homunculus. So homunculus just means little man in Latin, and what this demonstrates here, I think this is the motor homunculus and primary motor cortex, but there's a corresponding one in somatosensory cortex, which is just in the post-central gyrus. So the same principle is at work here. But basically the idea is that there's a large cortical patch of the brain that's sensitive to the face and also to the hands, as we see here. And for other areas, they have less cortical infrastructure to work with, to be connected to. So what this means is we have very high acuity of sensory processing for anything to do with the face or also the hands, which is what we need or what benefits us as a species. And so one way that you can detect this is there's a test that you could do. If you take a pin, for example, or a small needle, and you would blindfold a subject and you would tap on one finger or on different fingers, and then follow up with a secondary tap very close by but not exactly in the same place, and then again sometimes at the same place, and ask with those two pinpricks in the same place or in a different place, and you can gradually vary the amount of distance between those 2 points until people start to become 100% correct that you're able to detect a difference as well. And so because we have such large cortical infrastructure to quite sensitive areas, these are areas where we would be able to tell, even with perhaps a few millimeters, that okay, yeah, that needle prick is different from one that's very, very close by. But then if you repeat that with a poke at the lower back, for example, then you can actually poke the first time, and then the second time actually quite far away, and people will still say yes, that is the same space, because the mapping of the area, we don't require such sensitivity to we don't require such sensitivity in for our lower backs, for example. So that is why we're unable to detect consistently whether we are being poked in the same location, but we are for other areas of the brain that are very, very sensitive, and we need to have very high acuity of sensory processing in these areas. So cortical magnification is related to the idea of this sensory homunculus, and just like we saw there, there's like a distortion effect between what we're actually focusing on. So if you see here, if the eyes are focusing, or if the perceiver's eyes are focusing on the lady here, on her eyes as well, then you would see that the representation of her face at the back of the observer's face becomes distorted according to what we're actually perceiving, according to what we're actually focusing on, and what is the center of our fovea, and how is that information then being translated into representations back in primary visual cortex. And so here again we just see the same idea of distortion effect and the inversion. Now we're going to speak a little bit about hypercolumns and blobs. So as we talked about earlier, we have input from left eye and right eye. These are all sent via the lateral geniculate nucleus in both hemispheres back to the primary visual cortex. But what we see here are what's called ocular dominance columns, and ocular dominance columns are patches of cortex which accept information that has been segregated by the left eye and the right eye. And as you move along, you can see we have this recurring pattern of left, right in a very highly organized manner. And so what's on the left here is a description of some of the experiments of how we're able to detect that this happens. And along these ocular dominance columns, if we go in the other direction as well, we can actually see sensitivity to different directions of stimuli. So, for example, as we record from neurons slowly going up tangentially to the ocular dominance columns, you can start to see that there is sensitivity in an organized way to different orientations of stimuli. So Hubel and Wiesel were doing an experiment on vision in cats in the 1970s. And they had this setup where you used to have these old style projectors where you had different stimuli on the screen on this kind of small sheet of plastic that had stimuli on. You would place it onto the projector, and then the light from the projector would then project onto a screen, and they were trying to find out what areas of the brain in higher level visual areas that a cat would be sensitive to. So there'd been quite a bit of work already on the very basic level, and sort of like V1 just past there. So they were recording from slightly higher, and I think it was Wiesel that said that they were trying to discover sensitivity to different shapes, blobs that were on the screen, and the neurons that they were recording from, I believe his words were, they were frustratingly unresponsive. And then what happened was they were taking a break from one experiment, and as they were moving the stimulus sheet off the projector, as you did that, because it was like an A4 piece of paper and you're slightly moving it as well, what you actually end up recreating on the screen just for that slow moment in time is a oriented bar that would appear as you're taking the stimulus sheet off and about to replace it with another one. And then what actually happened was the neuron that was being recorded from in the cat was actually sensitive to a specific oriented bar at a specific angle, and that just happened to be what appeared on the screen as they were replacing the stimulus things to continue on with their experiment. And then they noticed, or they heard, that they whenever there's activity firing in these experiments, it is represented by a big crackling sound, so that the experimenters know that the neuron is firing rapidly. And this was the discovery that won them a Nobel Prize and set up a whole suite of future research that's helped us understand how we process complex shapes, and really got to the bottom of what are the building blocks that drive visual processing in the brain. And so back to receptive fields again. So receptive fields is, if you remember from a previous, here we are, we had a receptive field in white circle, and we had a light stimulus as well. And then when the light stimulus was presented in the receptive field of the neuron started firing, because this is what it's tuned to detect. Now we can have receptive fields in different types of neurons. So there are two further types of neurons in the visual system, called simple and complex. So we're first going to talk about the simple ones. And one of the features of the simple neurons is that they're called on center, off surround. And what that means is not just the fact that light has to be covering a specific portion of the receptive field, it has to be an absence of light in the areas around it as well. And so here we see firing rates. So we have on center and these off surrounds, and the receptive field is then this vertical band that you see here, and the bar of light that we present is then tangential to that, and this is why we don't see, well, we see a little bit of firing in this top bar, and then as we orient the bar of light to be closer to the aligned receptive field of this neuron, we start to see some more firing in the middle, which is to say, okay, there's a bit of overlap, some firing is going on in the middle, but there is something else going on in the outside. Then when we align the stimulus bar, the bar of light, with the excitatory receptive field here, because there's an absence of light at the side and light is present in the center, in the on center off surround neuron, we start to see this firing here. And this is a successful detection of a stimulus that the neuron's receptive field is tuned to detect. And we can see this again in this this picture to the right. So here we have the labeled on centers and then off surround, and we have baseline levels of activity when there's no stimulus in there as well. But if we present light going across in this way, we have the suppression effects from light being present in the off side. So the neuron wants to fire when there is an absence of light in the off center, and we actually have light present in the middle. So here we do have a bit of light entering the middle one in the central bar, but because there is no absence of light in the off part, this actually causes a suppression effect, which means we don't have any response in this neuron's receptive field. However, if we do present the light specifically where it is down the on center and there's an absence of light in the off surround, this is when we see very strong firing rates. And this idea of the on center off surround is very characteristic of the simple cell receptive fields. And so the simple cell receptive fields then lead to complex cell receptive fields. And so these are actually larger than receptive fields, and they do not have this on-center off-surround thing, and they're also what we call binocular, and that just means that if you remember we talked about the ocular dominance columns where we have input from the left eye and the right eye. This is actually something that only the simple cell receptive fields are sensitive to, but as we go and project to the further along the visual processing hierarchy to the complex cell receptive fields, then there's no preference for which eye a larger light stimulus, for example, might be tuned to. And one other feature that we have here is that some cells can be tuned to detect the directionality of stimulus. If we have here this yellow bar that has been moved over the receptive field, specifically in the left to right direction, then we can see there's a lot of firing of action potentials to signal that the thing that I've been tuned to, which is a vertically oriented bar moving in from left to right across this this region of, or this larger patch of the visual field. I have now detected this, but if we were to take the same bar and then move the other way, then we would still have something that only kind of looked like background rate firing and not really a true sensitivity to that bar that had moved along here. So the discovery of Hubel and Wiesel and these receptive fields and how we move from basic cells in the eye, so specifically these retinal ganglion cells here, which leading to the simple cells that down here, and then ultimately to the complex cells. What they proposed was that V1 was functionally divided into columns that are responsible for analyzing different inputs of the visual field. So if we were to consider a bar of light, let's say of this shape, so quite large, for example, and we were to present this to this retinal ganglion cell, we can see that it does overlap in the center, but then we're also providing light stimulus into the off surround. So if you remember, this causes the suppression of firing, so there is no response here, and then nothing happens down this pathway to the simple cell. And the same thing if we were going to present the same bar of light, again, nothing happens and nothing travels down the visual processing hierarchy. But if we did present it here, and there are a series of retinal ganglion cells that are together that would support the processing of detection of such a bar of light, then this would be contained primarily in the patched centers of on, and there would be no light stimulus in the off surrounds, which would lead to this strong response of maximal firing, and then we would see that this would lead to activity in the lateral geniculate nucleus, then down to the simple cells, and then further on from that. And so with that basis, we can now have a look at this top diagram here, which is the feature hierarchy, the hierarchical model that Hubel and Wiesel promoted. So here we have simple cells, and the simple cells lead to detection of the complex cells, and then we have hypercomplex cells here, and this gradually leads to an abstraction away from the detection of basic shapes in order to then support, for example, if you had a vertical bar and a horizontal bar and there was concurrent activity together, then this would be two neurons that fed into each other, and one would be sensitive whenever they were firing together, then that would be effective detection of a corner, and then from a corner, or various other combinations could lead to the detection of intricate shapes, and essentially this acts like the building block. So in the same way that we can make words of a language out of different characters, and characters are the basic blocks, then detection of basic shapes due to the specific organization of the retinal ganglion cells that then lead down to the simple and complex cells. This then acts as like the equivalent of the alphabet in recognizing shapes and allowing us to have the complex representations of visual input that we actually do have. And so the patch of cortex, the patch of grey matter on the outside of the brain that sits on top of the cortical sheet, is made up of a few different layers. So here we see layers, 1, 2, 3, 4, 5, 6. And then if you were to place a electrode recording, via say like in a needle, and you were going to go down into these different sub-layers of the cortical sheet, you would find, except for about layer 4 where there's something a bit unusual going on, you find sensitivity to the same orientation. So it's like a columnar organization where we don't see anything changing going straight down into the cortical sheet and our recordings. However, if we were to move along in the oblique or penetration, then this is where we would start to see sensitivity to various different orientations of stimuli, and it happens in a very it's not a random organization of orientations. It actually is as you move along, you would see the sensitivity slowly change in a way that is consistent and continuous. And so each cell responds to lines of slightly different orientations, and whether we're recording down in the brain or across the cortical sheet, we either see sensitivity to orientation across this direction, or more of a consistent response to orientation if we go vertically and record the electrical responses. So more about these ocular dominance columns now. As we saw before in one of the other slides, we have left, right, left, right, left, right, and this obviously comes through the optic tract from the retina to the lateral geniculate nucleus, and then these are projections going over to V1. And here in specifically layer 4C, where we have in red, we have left, and right. And this is how we get the ocular dominance columns, because of specifically the mapping of the information along the orbital radiation pathway from the lateral geniculate nucleus down to the primary visual cortex. And here we see a schematic of like the overall system, where we've seen that if there is a piece of information in the outside world, and light would hit that and then hit the retina, that information would then travel down the optic tract to the lateral geniculate nucleus in the thalamus, and then via the optic radiation pathway down to primary visual cortex, and then information then separates and propagates out. And so what we have here, a dorsal pathway and a ventral pathway. And so we can actually think of these as more as the what and where, or it used to be called the where, and now it's since more research has gone on into sensory processing, we now call it the how pathway. So it's not just where in space are these objects as you go dorsally up there, but it's more like how we interact with them. So what we actually see here is what we can call the what pathway, and then up here I'll just call it the how pathway. And so terms for you here are ventral, which is just like to the belly of something, and then how is more dorsal. So dorsal just means back. So if you're talking, for example, about a dolphin, people will talk about dorsal fin, a fin that's on the back, and as it's on the back, it moves upwards, and then ventral just means belly. So it's, if the brain was its own organism that had a belly, it would be at the bottom, and this is the origin of those terms. And there's a bit more information here about the specific delineation into a dorsal system and a ventral system and what actually happens, but we don't need to spend too much time on that. One other interesting feature of the magno and parvocellular pathways is that the what we call the M pathway and the P pathway is that the M pathway is what's sensitive to dark contrast, light dark contrasts. So it's kind of like the what the rods are sensitive to in the retina. And yeah, they're more sensitive at low spatial frequencies than high spatial frequencies, whereas the P pathway is more sensitive to color. So again, to draw that example back to the retina with rods and cones, we have rods which are responsible for light and dark, and the cones are responsible for color, and this is the same thing here. Information travels along the magnocellular and parvocellular pathways, the M cells and the P cells from the retina, and that is how that information is included in the whole system of visual perception. So that so far has been more about the biological underpinnings of how we take information from the outside world, how it enters the eye, and how there's more of a biological organization to it. So now I'm going to speak a little bit about more of a functional component, and one thing I'm going to talk about is this effect called prosopagnosia, which another word for this is called face blindness. So it's a disorder in some people that means that they're unable to detect faces. They can't draw, a sense of recognition from people. They have to build all of these unusual links between how they're able to recognize people, and for people who've had it for a long time, they just believe that everybody recognizes people in these ways, and it can only come out after quite a lot of time of suffering from prosopagnosia, where it might become obvious to either the person themselves, or it's usually the people in their immediate environment that notice this first. So one famous story is by this British neurologist called Oliver Sacks. Here's a picture of him here, and he wrote this book of the man who mistook his wife for a hat. So the story behind this was there was a music professor who's known as Dr. P, and his wife started to notice that he was exhibiting some unusual behavior, and it had to do with faces. So it wasn't just prosopagnosia or face blindness that he had. He also had a different condition that was more like pareidolia. He was seeing faces where they weren't there. So one part of the book that Oliver Sacks reports that his wife said was that he would pat fire hydrants on the side of the street because he would think they were children. And so the title of this book, like The Man Who Mistook His Wife for a Hat, is like Oliver Sacks was in the room talking to him, talking to Dr. P and his wife was standing by the door when they were ready to go, and he mistook his wife for a coat stand and put his hand on her face in order to pick up his hat. And then this gave rise to the title of the book, The Man Who Mistook His Wife for a Hat. And so the reason I'm talking about this is because what we want to go into a bit of detail in what are the brain regions that are responsible for processing stimuli at a functional level. So, what defines a place, what defines the perception of a body, what defines a face, for example. And one very, very commonly reported area in the brain that's been known about for decades now is it's called the fusiform face area that sits in the ventral visual stream. So if you see here, what we're actually looking at is we're looking up at the bottom of the brain. So here we would have an eye of one hemisphere, and here is the visual cortex, and we're actually looking up from the bottom to the top as well. So this is actually going to be the right hemisphere of a person, and then the FFA, and then close by this other area called the parahippocampal place area, one area is being strongly associated with face processing, and while the other one's being strongly associated with place processing. And as we're going to see in some of the paper presentations for this section, the jury's still out in terms of how specialized the FFA is for faces, and what other stimuli might drive responses to that, and what are the reasons for that. But basically, to get you up to speed in terms of a strong consensus that does exist in the literature today, is there's an area of the ventral visual stream called the FFA, which reliably responds to faces when presented in experiments. And here are some of the experiments that have given rise to that consensus. So here in each of these, we see a contrast between faces and another category of something. So we have faces versus houses, faces over scrambled faces, faces over objects, faces over hands, and this is again faces over hands, but with a slightly different measurement. And basically the point to be drawn here is that we're looking for activity that is recorded while participants are processing the first category versus the second category, and then you would subtract one from the other in order to see what is the net remaining effect of activity when faces are processed that is there but not there when processing the other category. And pretty consistently we can see here, we always see these responses to the FFA, so it's a very, very robust responder whenever you're presenting faces to people. One other mechanism, one other recording modality that you can use is electrocorticography, and this involves putting a sheet of electrodes, or a strip of electrodes, across a specific area of the brain. And this is what we call an invasive measure because it requires surgery to go in it. So it's considered unethical to do this purely for research purposes. But people who are suffering from seizures, they often they will require the placement of these electrode strips in order to help detect the localization of where the seizures are originating. And because of that, they're typically in hospital for many, many weeks, and when they don't have anything else to do as well, they're like a prime target for researchers who would give them a suite of cognitive tasks and try to record the responses from these electrodes. And typically when there's not much else to do in hospital, these participants are usually happy to agree, and this is how we get a lot of good intracranial electrocorticography intracranial data from for cognitive purposes. And here's one example of a guy who also agreed to be filmed as well. So this is about 10 years old, this video. He's an epilepsy patient, and he has one of these suites or strips of electrodes around the ventral visual stream as well, and so he has two electrodes over his face area. And we also have the ability not just to record from these electrodes, but also to send electrical stimuli and signals to activate neurons in that region as well. And we can observe what happens when somebody's processing a face, and then we purposefully interrupt that system, and we can try to get into what are the causal contributions of this. So if we send signals to activate neurons in the FFA, and somebody is processing a face and there is no interruption, then perhaps this is the effects that we see when presenting faces to participants in an fMRI scanner, for example, that this is just kind of a secondary effect of downstream activation. It's not actually a causally connected component, but the ability to interrupt the signal and see what actually happens during face perception. That's one of the things that was tried in this experiment as well. So I don't think I have the video available to play, but I have written some quotes from what he says. So he's actually looking at somebody else who's standing behind the experimenter, and he describes when they are sending the signals to the FFA. It's just, your nose went saggy, it went to the left. And then in the secondary one, when looking at a lady's face, he says that he had the perception that her face was stretched vertically. And, you know, he's like quite a surfer dude, and, you know, he's very very descriptive and hilarious in how he was reporting these effects back to the experimenter. Okay, so we're now going to finish with the basics of the human visual processing system, and then talk a little bit more about what's the inspiration for the visual system on modern neural networks, and the basis of vision models and how vision models work, also in a disconnected sense from how they work with in terms of human visual processing. And then this will then give us the tools that we need for the upcoming paper presentations, where these models are going to be used on a variety of data in order to come up with many different observations to drive forward our understanding of the visual system. So, as we see here, we have the nucleus, we have the axon, which gives an output. We have the cell soma here, and we have the dendrites that are connected here. We will go into more detail into synaptic learning in the reinforcement learning section, but right now we'll just leave this as pretty basic. That there are many inputs, and the strength of the connections are dependent on the dendrites, and we have this summation of activity, and if the resting state of the cell soma is depolarized enough to reach a certain threshold, and this triggers an action potential which leaves the axon hillock and then travels down the axon down to the end of the axon, where we have this release of neurotransmitters and synaptic neurotransmitter vesicles, which then are released into the synapse, and then promote the binding of those ions into the ion channels of the postsynaptic neuron, and this is how information is passed throughout the brain. And so the basics of neural nets that I know for the class that you will have already seen and done some basic machine learning, but here we have a set of inputs, and we can have a mapping to a set of outputs. And what we want to typically include is an activation function. So like the original activation function was, I suppose if you can ignore the step function that originally came with the multi-layer perceptron, the first real activation function we'll say was the sigmoid function and the hyperbolic tangent, or the tanh function. And here we see the sigmoid function and what it actually looks like on a graph. So depending on we would map anything from the real numbers along this line up to something in the range of 0 to 1. And at 0, we have a value of 0.5. But more recently, in more modern machine learning, and especially in deep learning, we have ReLUs, or rectified linear units. We have Gaussian error linear units, which are used in transformers and specifically in the GPT-3 implementation. We have Swish activation function, the soft plus, which is an approximation to the ReLU, and then also like the exponential linear unit and the scaled exponential linear unit. So there are many different activation functions, but primarily how they work is different depending on the task as well. And here's an example of a more complex neural net. So we still have our inputs here. We have 6 input features plus 1 that we prepend in order to take, in order to allow ourselves the ability to model the bias unit without having to explicitly model that. If we could just prepend a 1 to the inputs as well, and then treat it as an extra weight in the model, this simplifies a lot of the conceptual understanding of the mathematical implementations of neural networks. And so we can have this thing called hidden layers as well. So a hidden layer just means that we would have some inputs that don't actually lead to an output. we can have another hidden layer here. So the idea of the hidden layer is we don't really ever observe the values. We know what information goes in and we know what information comes out, but what actually happens in the middle during these intermediate calculations to get our final results at the end are not observed, and so this is why they've got the name hidden layers, or layers of hidden units. And so typically, say for example for H1 here, we have a weight for each of the inputs, and then these are all put through a specific activation function, and then at the end, this is our prediction, or this gives us our probability distribution over the outputs, and with the correct loss function, we can then calculate an error and then use the back propagation algorithm to update the weight in order to make the correct class more likely in the future when seeing the same or similar input. And so in convolutional neural networks, which we're going to look at in a little bit more detail later, this is an example of features that are learned by convolutional neural network. So what we actually talked about in terms of the patches of the visual hierarchy that become sensitive to various orientations of shapes, especially with these ideas of like on, center, off, surround, similar. So you can see here that there is some sensitivity in a lot of these where you have white and then you have these black bands next to it, and these are specifically to detect sharp corners and lines, and this would be from a very, very early layer of a convolutional neural network as well. And this wasn't inspired by the brain in a basic sense, but the fact that we can recover these activations from there that seem to build up the same alphabet of basic shapes in order to give rise to the modeling of more complex shapes it's been a really interesting idea that led to a few questions about exactly what's going on in terms of when neurons we're training convolutional neural networks, and what are the similar representations that might be coming from the brain. So regularization in machine learning is an important concept because we're learning with a much larger number of parameters, and this makes models very prone to overfitting. So what is overfitting really? If you imagine that we have these data points, we have each one of these that I'm highlighting here is its own data point with an x and a y value, and this is what we're observing, but it's a very, very noisy data set. So if we imagine that the true data is should be this line, but we're actually having a very noisy measurement of that system. And so if we have a very, very complex model, the model would want to try and be trained to predict each of these points almost exactly. And then so we can imagine if we are going to get another sample from the same system and it's going to be noisy as well, it's very likely not going to be the same, or with a good test error, to a model that's been heavily overfit to a series of data points that have a lot of noise in them. So if we apply regularization, it means that we're not sensitive to the extremes and the outliers. We just want the general trend here. And this means if we do see a point in the future that might be here or here, we're actually going to be able to better model those points after training the model by including a lot of regularization. So dropout is one method of regularization, and what this involves is randomly dropping connections in the network, or specifically as we might implement it, it's setting the weights between them to be 0, so actually no learning signal can go along towards the prediction or then come back towards the output as well. And this is kind of like an ensembling procedure, because at each iteration when we're putting different information through, we're randomly turning on and off different neurons, and so we break up this ability of neurons being able to overfit. So for example, we might see specific neurons always trying to model very specific inputs and not general trends that exist across the data set. But if we are randomly turning neurons on and off, especially if we have a high dropout percentage, we're actually forcing the model to pay more attention to the generalities, and then we would say ensemble the results at inference time. And this results in a model that is more capable to generalize and has a strong regularization effect as well. So here, if we're looking at classification error on two models where we have without dropout and with dropout, we can see that when we've applied dropout, as the number of weight updates increases along the x-axis, we actually have less classification error when we are using dropout compared to without. So not only are we avoiding overfitting, and we're actually helping the ability to the model by forcing it to concentrate on more generic concepts that are more useful for learning. We actually have a better result overall. So dropout, as we can see, we have a model without dropout, which is just like what we would call a fully connected network, and fully connected because each neuron is connected to every other one. And we start with the input layer, we go to the hidden layer, and then we have a binary classification at the top here. But with dropout on one specific training iteration, say we would turn off this neuron and this neuron, so all those connections are broken. From you don't see these non-existent to the dropout, the dropped layers and the hidden layer. And then this means that the information that's being passed from the input layer to the classification layer is going to be different each time, and therefore no one neuron, because each time you're being trained you have a model of reduced capacity, it's actually less prone to overfitting because it has less capacity, but then we allow for more capacity by allowing other general concepts to be learned. And so we get to convolutional neural networks. So these are built to capture the invariances we see in image, and so what we see here is that if we have a model, we don't want to specifically build a model that detects a mug of this shape in this specific location. We would like a model to be able to learn, what is the concept of a mug, and then also be able to detect it in many, many other different shapes and sizes, and have it as an effective feature detector. So unlike the retinal ganglion cells in the brain, which are only fixated on receptive fields at specific locations, as the visual processing hierarchy in the brain gets more complicated, we actually see these invariances to location, and so the detection of any complex shape in any part of our visual field would eventually lead to a signal being passed up where, as I said, it wouldn't really matter where it exists, where it appeared in our visual field. The detection or the response to say this object is in my visual field somewhere, that is the case, and we want convolutional networks to be able to do the same thing too. And so how they work is they learn filters that are that respond to particular visual features, so like edges or curves, what we saw in this map here. So this is like a set of feature vectors that are used to detect various basic shapes, because this would be from a very early layer of a CNN. And we would be able to find those features anywhere in an image. And so what we do with these filters that are learned in a CNN is that they are convolved over the image, and we progressively update and apply various extra steps such as pooling in order to build in some translational invariance into the model, and ultimately propagate the output of the kernels, which are together combined to be responsible for detecting low-level and high-level information, that they need to send the signal ultimately up to the top of, to the output layer. So yeah, the hidden layers combine with these filters to create complex shapes. And so a quick history of CNNs. So first of all, we need to talk about the Imagenet data set. So the Imagenet data set is this giant data set that was released as part of this challenge to the computer vision community in 2009, which contained more than 1,000 concepts and 1.2 million images. And they're very, very complicated sets of classes. It's not just very basic things. It's very, very low level classification information that I think most humans wouldn't be able to achieve 100% on. So here are some samples from the Imagenet database. So we can see up in the corner here we have, a lot of butterflies. Also around here, many different animals, lots of buildings, scenes and boats, etc. And here's an example of some of the 1,000 classes in the Imagenet dataset. So we can see some trees. Here, so we have fringe tree, European ash, the New Zealand beech, live oaks, shingle oak, cork oak, yellow birch. As you can see, this you'd have to be almost an expert at forestry to be able to do as well as a model that would get 100% on all of these tree classes in Imagenet. We also have many different visualizations of categories in Imagenet. So for example, it's not always a cat in the same position, perhaps tail up or looking. We actually see cats lying down, filling up the space of the image as well, sitting on a bed without backgrounds, occurring with humans or multiple ones as well. So it's actually not a restrictive sense of if you're going to include a category, then all images would be the same. We actually need to model a category. They all have to, all of these images have to be detected as cats, and because we see such a variety in terms of species of cat and their environments and how much they fill the screen as well, the model really does need to learn general concepts about it and things that are common to all of these images, and which really gets to the heart of learning or having the ability to detect what is a cat in an image. People were using traditional computer vision approaches and models from 2010 to try and tackle this. And here we can see we have the accuracy up along the y-axis. And so in 2010 there was a pretty big range of models that were achieving various levels of accuracy. In 2010, then we get to 2011, we see these models that worked for the previous iteration, the ones that were below 40%, for example. People have abandoned, people started to take and tweak the successful models from the year before. And then we also see in 2012 that this is when AlexNet model was created, so by Alex Krzyzewski, Ilya Sutskever and Jeff Hinton. And what we see here is people were using the traditional computer vision models in 2012, but then we see the arrival of the first CNN when it was submitted to this task, and we start to see that it beats the performance of all of the other models as well. So it's the first one labeled in green here. And then as you see, pretty much after that, people were so surprised that CNN models were so able to successfully model the classes of Imagenet that by the next year, most of the implementations were some form of CNN or adaptation of the AlexNet model. But still, there were a few maybe people with the lowest performing here that was submitting the traditional computer vision approaches, and as you can see, compared to the year before, it was actually only the very, very top level ones that were carried forward. By 2014, people were only using CNN. And so what kind of implementations can CNNs help you with? So one is with self-driving cars, for example, or here specifically semantic segmentation. So if you see, we have the street scene here, and the task of the CNN is to be able to color code each pixel in there and label it as, to give it a label in order to be able to map out something in a more useful for downstream applications. So, for example, we see these red patches here. These correspond to the buildings, which we can see here. And then if we see this grey over here for sky, all of this patch over here. So it's effectively abstracting away the information from what is actually the detailed stimulus in order to say, okay, please split this into what is a vehicle, what is a fence, what is a tree, pavement, road, marking pole, etc. And one main application for this is in self-driving cars. So if we have a camera at the front of a car and it needs to be able to split the actual image that's been taken, which we see on the left here, and then divide it into what is actually another car, what is a potential a road marking sign, a pedestrian, a bike, for example, and be able to use this information in order to more successfully learn how to navigate and be sensitive to potential danger areas. So, for example, if there's the case that there's another vehicle in front of you and it's designated by the specific color, and it becomes dangerous, then the model would know, okay, I need to take this action in order to not cause an incident, to not cause a crash, for example. But yeah, back to two additional operations in CNN. So they're like the convolutional operation and the pooling or the subsampling operation. So we have this input here as a robot. Then we define how many feature maps that we want to learn, which are specifically defined by smaller kernels. So we would run a specific set of kernels over the input, and then our output would be a set of feature maps. Then we do a subsampling procedure, and then we would have a smaller set, which we then treat as an image with many channels, many in addition to the 3 color channels of the input, which we see here, because this has this is a color image, so we would imagine a channel for red, blue, and green. Then we would run an extra set in a higher layer of the CNN. It would be a new set of feature maps, and they would apply to the previous layer, and then we would do subsampling again, and then we would eventually get to a fully connected layer, which then maps to the output class. So we're taking these small detectors, which are gradually getting more complicated the more layers you go up through the neural network, and then we're applying pooling operations, which I'll speak about in a few slides. So although some of the images I've been showing you previously, they actually look how we expect images to look. Basically, if you just zoom in very, very, very close, they're just numbers in a grid, numbers in a matrix. So here, if we have an image which in which we depict either binary values, either 0 or 1, then basically we're going to call this thing here, this green matrix, an image, and it has values in it. And then we have a smaller filter which also has values in it. And what we then do is that we then take the Hadamard product of applying the filter onto an image and then storing the summation of that value, and then moving along the filter across the image until it covers the entire patch of it, and we're always recording the output of the Hadamard product and the summation in what we call the activation map. So a typical filter might look something like this, and for color images in the bottom we can see a few here. We can also see some from the top are basic levels, like low level CNN filters, which are detecting orientations of specific shapes. So again, this is what we actually see in the brain down in V1 and low areas that need to either detect various primary distinctions between orientations and shapes. And then in the second block here, so I'm just highlighting here, this is more of a high level. So the learned filters from high levels of a CNN that have more to do with color and more abstract shapes, like this kind of checkerboard thing that you see here. This comes from the AlexNet paper. So what we see here, we have a filter here, which we're going to represent by this small filter here, and we're actually going to iteratively apply it over the image, and after that we would get this image, which has abstracted away a lot of the detail that the actual filter doesn't really care about. You can see that this filter is sensitive to edges, which are important in an image in order to derive a lot of shape features. And so then this is our final, say, feature map, which would then be fed into a subsequent layer, and then with potential like subsampling or pooling operations on top of it in order to condense this information a little bit more. I think this bottom one is actually the response to a slightly different filter though. And here we arrive at the pooling of the subsampling layer. So here is the same robot network that we saw a few slides ago. And here, if we zoom in on what's going on in this image, just by this building, if we zoom in, we can imagine we have the output here of the filter being applied to the image at the step. And so if we do want to apply pooling or subsampling, then we have to figure out what is the value that we want to take forward in order to reduce the dimensionality of this representation. You know, we can either take the max, which here would be the value 4, which then ignores a lot of other information, if that might be relevant. We did use the max for quite a while, and the rationale used to be that you would only get strong responses from a filter. When a filter was applied and you would onto an image and you would get a high response, that is the thing that that signals detection, and the highest value is the most important one. So we would take over the maximum one. But then, you know, there were various cases where that wasn't ideal, and people thought, okay, what about if we just take the average? And you know, the absence of other parts of that filter, especially if they were larger filters, would also help play a role. And so the reason that pooling or subsampling provides some sort of invariance is because if you imagine quite a large filter, and we go back to, say, the coffee cup modeling images before, if you were to apply a filter over a specific image of a coffee cup, and let's say this filter was designed to detect coffee cups or cups in general, then if we were to just take a single value out of that detection to the highest value, and then move that up to a higher layer, then we would know that there was a detection of a cup, but not at the exact location of the pixel value, because we've only taken either the maximum or the average, and we gradually lose that spatial information, which we don't really mind, because if we just want to detect what is actually in an image, we don't actually want to go back and say, and then here is where we find it in the image, then this is why we can talk about having this like local invariance, and you know, the ability for a model to not need to be so sensitive to where exactly it finds whatever the kernel is tuned to find via training. And so the feature representations that we find in the middle of a network, which are like the hidden layers, which we talked about. So we have like the classification layer, or the output and prediction layer, and then the input layer, and then what we don't actually see the values of in the middle are what we call hidden layers. And so the output of the feature maps before, and when we go through these pooling and subsampling operations, these effectively are the hidden representations of convolutional neural networks. And so we'd see it as a compression of information, and not unlike principal component analysis or singular value decomposition. And so here we can ask, what is it the CNNs learn? So we've seen variations on a few of these figures before, but here we can see it's a variation. This doesn't look to be specifically low level. Maybe it could be from a middle layer of a CNN as well, where we're detecting all sorts of bends or circles or crosses here. This is basically saying that it's like sort of being given a language and said, okay, give me the alphabet that will help me model this language in the future. But in terms of the visual language, it's saying, what are the best component shapes, or how can I take the structure of the visual input and break it down into like a basis set of what's going on in relation to these shapes that have been learned, and that is what we find as that CNNs learn, especially in different layers. We see from the basic layers that are closest to the input, we find more ordinary geometry in terms of straight lines, and then we find much more complex things the further higher up we go in the CNN hierarchy, in the hierarchy of layers from input at the bottom to the classification layer at the top. So this is from, this is like the eminence data set. So the eminence data set is a set of numbers between 0 to 9, which was originally designed for the US Post Office in order to automate the detection of numbers on post that was passing through the post office. And so while the CNN was trained on this eminence detection task, these are the features that were learned that the model thought would be best in order to model all of these classes. In order to be able to successfully model the detection of like handwritten digits, we would find a lot of these circles and a lot of these like holes and gaps and little shapes here that together allow, so these are the set filters that then go over a specific image and then are useful at detecting the various components of handwritten digits. We'll talk a little bit about adversarial examples now. So we can make small changes to an image which are like almost imperceptible, and then this causes the network to misclassify. So this is actually, although we've talked about very large similarities between what are the shape detectors that happen in early visual cortex in the brain and then what happens at lower level layers of hidden vector representations of, or what is the visualization of kernels in lower levels of CNNs, and there is a pretty strong similarity between what the brain does and what those CNNs seem to do. But we actually know that the models and how they're affected by adversarial samples are very, very unbrain-like. So we'll see a few examples of this now. So what we have here is two cases, 1, 2, where we take the first 3 inputs, we would add a layer of distortion, and then we would ask the model, okay, predict for us again what are the classes here. So we have school bus, we have this bird, and then this temple. And we add these 3 different layers of distortion, and then the model now thinks all of these is an ostrich. And in the second example, we have this speakers, is it? And then a stick insect, and then a dog. We add these distortion effects, and again, this is how we make the model always predict ostrich, because it's sensitive to this imperceptible, very, very small difference that has been added onto the original image, specifically designed in order to make the model misclassify the inputs as ostriches. And we can also put in images that are very, very like noise as well. So this first one here, the model detects as a robin. This one is a cheetah, armadillo, and a lesser panda. I don't know if I'm just imagining it because I've seen the label first, but there's something quite robin-like about this thing, perhaps not. And then also we can put in some crazier shapes and see, okay, what does the model think these are? So like king penguin down the bottom, starfish, baseball, electric guitar. We can see that there's the way that we've set up the models to force them to make a prediction, and where those decision boundaries lie for each classes means that they're forced to make a prediction, and can be very, very confident about them. So for here, this is the estimated over 96% prediction probability that these actually are the classes that are present in these random nonsense images. So one way, instead of looking at the output of the feature maps in order to say, okay, what is the filter that will be applied over an image in order to detect a specific characteristic of that image, we can consider a specific neuron in the input layer of a CNN, and also one of the values from a feature detector in the middle of the network, and we can say, okay, how can we change the input in order to increase the firing of the specific neuron? So basically, we can take the gradient and we could say, okay, what changes do we have to make to the input in order to make this neuron fire the most? So this is very different from what we've done before. Usually, when we're making an update to the weight, we're always updating the weight of a neural network. Now, what we're actually doing is we're taking an input and we're changing an input in order to make a neuron fire at a higher rate. We could phrase it like that. And so one of the papers that we're going to see in the upcoming paper series is this paper by Horikar and Kamitani. And they took this idea and had very, very interesting observations. So they also had this 8-layer CNN we have in A. We also had a set of comparison images, so the HMAX model, GIST, and to sift and bag of features. And so here we can say we put in an image of a chimpanzee and we get the models. They also recorded fMRI activity when humans saw the same images, both in a sense where they were actually given it to perceive freely in the scanner, but also when they were imagining it. So this whole paper rests on the experimental analysis of decoding imagined versus seen features. And so our model was built by having the person look at the fMRI activity and then training a decoder to look at the distribution of the patterns of the neurons and responses, and then look at these category averages across the data set in order to derive these feature patterns, and then pick the closest matching one in order to do brain decoding of both seen and unseen features. And so these were kind of more of the control models that you see here, but the model of interest was AlexNet, I think it was, and they then looked at the learned features and then what they did was they looked at they took the gradient of the feature maps neurons in different layers and then asked, okay, what is the input that drives the maximal responses to these neurons as well? And so as you can see here in CNN1, so exactly like with the retinal ganglion cells that we saw earlier, neurons here in the first layer are only sensitive to information at very, very specific shapes, spaces in the visual field, so only here, and there's obviously a very strong color dimension here. And then as we have the subsampling and pooling operation, then the receptive fields of neurons in CNNs gets progressively larger, exactly like they transition from the receptive fields of simple cells to complex cells. And so we start to see some crazier patterns here. So based on the the inputs, we start to say, okay, in the second CNN layer, we start to see something that looks like a bit like an eye here, and then these patterns start to get a bit crazier. We see it in CNN level 3. Again, the receptive field becomes wider, and we start to see the modeling of more unusual patterns. So this is saying responses like this, they were never seen in the images that were input into the model. This is that the model were trained on naturalistic images, but then now we're artificially generating inputs to say, okay, what would be the input that if we gave this to the network that would give you a higher firing rate, and so what are you most sensitive to? And then we can go further up to CNN level 6, 7, and 8. As you can start to see that we have receptive field is no longer restricted to a subspace. We can actually see this tiling of patterns because of our local invariances. And we can start to see, okay, there are some things that look like animals here. This looks like shapes of a vase or something, and then here something looks like a cat or a dog, or animal-esque at least. And then here in CNN7, for example, we can definitely start to see something that looks like animal shapes around here. And then by the time we reach the highest layer, we actually know what the output is. So because each unit of the output layer is tied to a specific category classification label, then we're able to say generate the input that is most likely to trigger the activity to make this specific category be present. So we would actually expect that the artificially generated inputs should look something like the category of the class that they were specifically designed to model. Obviously not in terms of a realistic image, but by being able to detect the generalities of the class that it wants to detect. So basically, if there is this coffee cup, for example, on many different types of tables or in many different situations, we don't actually really care about the background. We only really care about the representations of that, and so we would only want to see the representations of the shape for how we might expect a model might need to model the features of something like a coffee cup. So here for vulture, we definitely see something like birds, and gazelles definitely see this, and mushrooms and cups as well. So that's the end of the vision lecture, and we're going to begin the next few weeks with the paper presentations, and so good luck with that.